Hello, this is a summary of a project entitled Exploring Biodiversity with First-Year Biology Students. My name is Bridgette Clarkston. I'm an instructor at the biology program at the University of British Columbia. The undergraduate course involved in this project is called Biology 121, Genetics, Evolution, and Ecology. Biology 121 is a large lecture-based course with no lab or tutorial, intended mostly for students who will eventually major in biology, but also taken by many other science and non-science students as an elective. Each semester I have roughly 160 to 190 students per section, and there can be up to 7 sections running each semester. I want my first-year biology students to gain some understanding and appreciation of biological diversity, including how biological specimens are collected, preserved, and cataloged. So with this in mind, I partnered with BLUE, the Biodiversity Literacy in Undergraduate Education Network. Through this network, I revised a project I developed back in 2017 with my UBC colleagues Linda Jennings and Pam Callis. This project asks students to put on their citizen scientist hat to help document biological specimens from the Beaty Biodiversity Museum located on the campus of the University of British Columbia Vancouver campus. The Beaty Museum houses all of the UBC biological specimen collections, with over 200 specimens in total. These include cases and cases of mammal bones and skins, jars of preserved fish, fossils, dried plants, crusty seaweeds on discarded bottles, even an entire blue whale skeleton. For this project, I partnered with the UBC Herbarium, the section of the Beaty Museum containing all plants, algae, lichens, fungi, and bryophytes. It contains roughly 750,000 specimens. Like many other museums around the world, the Beaty Museum is in a process of digitally photographing all of its specimens in order to make these specimens available online to both the scientific community and the general public. Along with photographs, the information about where and when a specimen was collected found on each specimen's unique label must also be transcribed digitally. And that's where my class made their contribution in this project. I have 5 specific student outcomes for this project, and I should say the entire project was completed by students outside of class time and through digital materials they were provided. They were not required to visit the museum. Although they were encouraged to do so, the museum staff cannot handle hundreds and hundreds of students coming in to contribute to a project in a single semester. The first of 5 outcomes on this project was for students to explore variation within a single species. Because UBC Vancouver is a coastal campus, and I personally am a seaweed nut, I always choose charismatic seaweeds as the focal organisms for this part of the project. Here we learned about the iconic bull kelp, Nereocystis lutecaena. Students were provided multiple high-resolution scans of pressed specimens from the UBC Herbarium. They then completed a worksheet prompting them to think about variation between the specimens. A portion of the worksheet is shown here. The next 2 outcomes taught students about collecting and preserving biological diversity. Students were provided a slideshow summarizing background information about herbarium collections, including just what a herbarium actually is, how specimens are collected and the adventures therein, and about what information is typically included along with a dried herbarium specimen. Another worksheet accompanied this part of the project. Again, a portion is shown here. In the 4th outcome, students completed digital transcriptions of Beaty Museum specimens. Like many other museums around the world, the Beaty Museum is working to digitize its 2 million specimens so they may be widely accessible to researchers and the general public. This enormous task, however, requires more time than museum curators can spend typically, so there are many cabinets full of specimens that have never been photographed or digitally transcribed. So for this part of the project, Linda and her museum staff use the citizen science platform Zooniverse, where they upload photos of herbarium specimens. My Biology 121 students then go to this website, see the specimen photographs, and type out the information on the herbarium label. The Zooniverse platform provides a user-friendly interface and customizable tutorials that train students in providing accurate, precise transcriptions. As simple and almost tedious as this work sounds, students reported really enjoying this part of the project. When talking to students, the general sense is that they appreciate doing something that contributes to a scientific project beyond their own classroom, and no student does more than 3 or 4 of these transcriptions for their own project. The final outcome of this project is for students to explore biodiversity data once it has been transcribed and made publicly available using an online biodiversity database. I chose to use the Consortium of Pacific Northwest Herbaria website because it's an easy way to access UBC herbarium collections and to see their distribution on a map. Shown here is an annotated screenshot that I use when providing instructions to students for doing their searches. Again, students completed a worksheet prompting them to search for local BC species, explore their distributions, and generate ideas for future research collection events. In terms of results, the number of students for this past semester were 169 in the course. They completed a total of 825 transcriptions. And since each specimen must be identically transcribed at least 3 times before it is removed from the Zooniverse system, a total of 265 specimens were completely transcribed as part of this project, for an average of about 3 specimens per student. As part of this project, students also completed surveys before and after the background information and specimen transcriptions to explore their perceptions, values, and understanding of biological collections. In the pre-survey, we asked them what they thought, why they thought, or if they thought more than one specimen from each species needed to be preserved. And a common theme across many students was that no, a second specimen is not necessary, or if it is, it's just there as a backup in case the first one is damaged. We also asked what they thought was included on a specimen label before they did their transcription part of the project. And again, there was a wide variety of information provided. Often they knew the scientific name should be included, but other things like vague background information or DNA or carbon dating or ecosystem roles were commonly put as well as information that should be included on a specimen label. We specifically had the worksheet questions targeting what information goes onto the label so that by the end of the project, students had a more accurate understanding of what information is collected when a specimen is collected. In the post-project survey, one question we asked of the students was, what is an additional question you have or curiosity you have following this activity? And the students reported all kinds of really enthusiastic things, like wanting to know if they could participate in research, wanting to know what happens to these specimens after they're preserved and digitally transcribed, and asking more sophisticated questions in the post-survey than in the pre-survey. Things like, who is responsible for deciding what is preserved and what is not? And what exactly goes into preserving or deciding what collections are made? This concludes the summary of this project. I want to thank Blue, the Faculty Mentoring Network specifically, for encouraging me in this project, my collaborators Linda Jennings and Pam Callis. And if you're interested in these resources from this project or a description of it, it can be found on the Blue Cube's website, linked here on the slide. Thank you.